Presentation will be from Miss Sani Hanifah. Miss Sani Hanifah, maybe you could start share your screen. I would first provide the profile about Miss Sani Hanifah. So Miss Sani Hanifah has over 15 years of experience in training and teaching agricultural human resources. She is an agronomist. She has extensive professional experience in horticulture and tissue culture. And as part of tissue culture technology, she trains in tissue culture media. She is now working as lecturer in the Indonesian Agricultural Training Center of Lembang under the Ministry of Agriculture, Republic of Indonesia. Ms. Sani holds a master's in agronomy from University of 11 Maret Surakarta. And in this session, she is going to present a topic about tissue culture media. Ms. Sani Hanifah, please, the floor is yours. Thank you.
Okay, today I will explain about tissue culture media. What is the component of tissue culture media? There is 8. The first one is distilled water, and the second is macro and micronutrients. Also, we need sugar, vitamins, amino acid, plant growth regulator, compactor, and activated charcoal. Okay, distilled water we use as a solvent. For making stock solution, we must use distilled water because it's no longer contain mineral and organic compounds. Okay, the second is macro and micro elements. The macro elements are nitrogen, phosphorus, potassium, sulfur, calcium, and magnesium. The micro element is iron, copper, manganese, zinc, boron, molybdenum, and cobalt. Elements that are needed in large quantity we call macro element, and in a little we call micro element. Okay. And sugar. Sugar we need for energy source. The standard source of carbon are sucrose and glucose. But the most widely used for tissue culture is sucrose. But if it is difficult to find, we can use white sugar that we use daily. This white sugar is also qualifies for culture growth. And we also need vitamin. These vitamins are commonly found in plants, but other vitamins that are often added to tissue culture media are niacin, glycine, myo-inositol, riboflavin, biotin, and etc. But myo-inositol is often added because it helps to cell division process. Okay, and the 5 is amino acid. Amino acids are source of organic, which is taken up more quickly than inorganic nitrogen in the same mediums. Amino acids play an important role in callus growth and differentiation. And 6 is plant growth regulator. Growth regulator in plants are non-nutrient organic compounds which in small amounts can support, inhibit, and change the physiological process of plants. Therefore, growth regulator in plants consists of 5 groups, such as auxin, gibberellin, cytokinin, ethylene, and inhibitor with different characteristics and effect on physiology. Okay. I will explain about auxin. Auxin inside the plant body have a very complex effect and can control growth. The physiological roles of auxin in plants such as for enlargement of cells and organs, root formation, maintains apical dominance, to stimulate vascular tissue differentiation, also trigger ethylene synthesis. There are 2 types of auxin. Natural and synthetic. The natural we call indoleacetic acid, but synthetic is indobutyric acid, naphthalene acetic acid, and 2,4-D. This is the role of auxin in tissue culture. The first is to inducing roots. Also for induced callus, induced somatic embryogenesis, and increased ethylene synthesis. Auxil asynthesis at tips of roots or shoots. Okay. Here is cytokinin. Cytokinin are the name of growth regulators that are very important as a stimulant for cell division and morphogenesis in tissue culture. More than 30 types of natural cytokinins, but two are widely used in tissue culture as zeatin and 2-EP. Synthetic cytokinin are kinetin, BAP, and BEP, and pidiazuron. Okay. In tissue culture activity, cytokinin has been sought to stimulate the occurrence of cell division, callus proliferation, formation of shoots for flowering, chloroplast formation, and inhibits root formation. Here is gibberellins. Gibberellins can affect the elongation of the stem or stem segment. Also encourage flowering, fruit induction, and breaker of bud dormancy. The growth regulator of gibberellin we use in tissue culture is GA3 or gibberellic acid. And the seventh is compactor. The compactor that is often used is agar. The advantages of using agar are in order to freeze at 45 degrees Celsius, and it can melt in 100 degrees Celsius, so that in the culture temperature range, the agar will be in a stable frozen state, not digested by plant enzyme, and does not react with compounds media. The last one is active charcoal. The active charcoal is a charcoal that has been heated for several hours using steam or hot air. This material has a very strong absorption power. Effect of adding activated charcoal to media, it can absorb toxic compound which can inhibit the growth of culture, especially the phenolic compound in tissue injured at initiation. Also, it can stimulate roots by reducing light reach the exposed parts in media. And this is a buffer or pH media. The pH must be adjusted in such a way that it does not interfere with the cell membrane function and the pH of the cytoplasm. Plant cells require a slightly acidic pH ranging from 5.5 to 5.8. The pH adjustment is usually done by using sodium hydroxide or hydrochloric acid. When all the components have been mixed, sometime before being sterilized in autoclave, the addition of amino acid is often an organic buffer. Okay, thank you for your attention. If you have any question, me and my colleagues can answer your question.
Sani Hanifah, thank you for the comprehensive presentation. There are one question from Walid Kaya, Lebanon. Maybe you could see it in the Q&A feature. Maybe you could respond to his question.
To Walid Kaya, you can make yourself a media from we can use the instant Murasige Skog media. You can buy in chemical store or online shop. So you not have to weigh one material. You can use instant media. Okay. But the price of media, the instant media, is quite expensive. So we made macro and micro element and we can prepare ourselves macro and micro elements more cheaper than we have to buy instant media. Okay. Yeah, thank you, Walid Kaya. Okay, for Mokhtar Hussain, which part of plant would be suitable to produce disease-free plant using this technique? Which plant? Masih media belum gitu. Jadi apa? We can use meristem site to produce disease-free plants using this tissue culture technique. Okay, Dr. Mokhtar Hussain, we can use meristem. Thank you, Mokhtar Hussain.
Okay, Dr. Mokhtar Hussain, are you satisfied with the answer from Ms. Sani Hanifah, or would you like to? Okay, I think so there's one Ms. Sani Hanifah, there is one participant. Oh, Dr. Mokhtar Hussain apparently want to address feedback verbally.
Okay, yes.
Dr. Mokhtar Hussain, yeah, okay. Please speak.
Hello.
Yeah, hello.
Can you hear me?
Okay, I can hear you.
Yeah, sorry, I joined with you a couple of minutes later, and also I faced some trouble from internet connection. Therefore, I couldn't hear all part of this session. In fact, this part is fine. I hear you very clearly, and I saw your presentation. It looks fine, and it will be useful for the those who are attending in this session. And one of my requests is that you can send us this, if possible, you can send this file to all the participants so that they can, or we can try to apply these techniques in our labs. That's it.
Yes.
Okay. Maybe yeah.
Okay, maybe for, okay, I'm sorry. Maybe to Dr.
Mokhtar Hussain.
Okay, to Dr. Mokhtar Hussain. My name is Fia, and I'm going to help my friends to a little bit give a clarifications about this. Actually, for all the materials and all the video contents, actually you can have them all on the LMS, and yes, maybe you can practice after the sessions, and all of the material are already provided. So due to the problems from some of the participants that cannot access the system, we decided to play the videos in this Zoom meeting session. But actually, you can have all of the material, including the module, in the system.
Okay, thank you.
Thank you very much, Dr. Mokhtar Hussain. I think the organizers will share all the presentation files, including the YouTube link for the videos on the technical procedure about the tissue culture media.
Yes.
Yeah. So there is, I'm sorry, Ms. Sani Hanifah and Ms. Viadini, there is another participant who would like to address his feedback verbally from Mr. Buddha, BK from Nepal.
Okay.
Hello.
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 Hello, very good morning and namaste from a beautiful country, Nepal. Myself, Buddha Vike. I have participated in this Zoom meeting and I got lots of knowledge regarding this stock solution and media preparation. While at a presentation time that you have shown that one video for stock solution preparation and other by side, there was in that video I have noticed that the stock solution, pH of stock solution should be at the range of 5.8, and while at the presentation it was shows that the pH of stock solution may be or should be at the range of 5.5 to 5.8. My concern is that it will be fine if the stock solution pH should if it have from 5 to 5.5 to 5.8, or it should be 5.8. Thank you.
Excuse me, there is a little bit problem with our device here. Can you repeat shortly about your question?
In brief, my question is that not a question, it's my theory that
Question.
is the pH solution of pH of stock solution should be 5.8 exactly, or it should be at the range of 5.5 to 5.8.
Oh, it's about pH solutions, Miss Sani. It's about a pH solution.
Oh, pH solution. Oh, pH on a media. If we're making media and after the distilled water, micro inside the beaker glass, and we add sugar and we put a pH. If the media is less than 5.8, we can add some sodium hydroxide. But if the media more than 5.8, we can add hydrochloric acid. That is pH solution for media. And after media 5.8, you can add agar and then you boil the media. Okay. Okay, Miss Via, can add some more?
Okay, maybe because the range of the pH of the media is about 5.5 to 5.8, it's also related with the availability of the nutrients. So if the pH of the media is above or lower of this range, there is a possibility that the nutrient is not ready for the plants.
Yeah.
That's why we need to adjust the pH of the media to make sure that the planets will get, or the eggplant will get the nutrients from the media itself.
Mr. Roga, do you have a further feedback about from response from Miss Sani and Miss Via? Mr. Buddha? Okay. So,
Thanks. Thank you. Thanks for. Thank you.
Thank you.
Okay, thank you. So, Miss Sani, there is another question from the Q&A from Reski Yulianti.
Yes.
Can you tell us the name of the instant product? Can we use PGA media?
The instant product we use is Murasigas media.
We will show you.
We will show you.
Okay.
Yeah. You see? Murasigas. Yeah. One media for all plant land.
It's already on the package, so you don't need to have to wait one by one of the nutrient is already included in this instant media, so you can use it directly to make your tissue culture media. Yeah, you just only weighing and pouring into the baker glass and also add the sugar. Add the sugar. So that one is the instant media that you can use.
Untuk bakteri. For PDA, we use two fungus. Potato dactylus agar. We use media for fungus. Yeah. Miss Resi. Okay, thanks. Miss Sangeeta Devi, thank you. Could we use normal bottle cap instead? Oh, okay. For Miss Sangeeta, yes, we can use rubber capstock or we use plastic for cap stock solution. Oh, for stock solution. This one.
Maybe I can show you. This is for the stock solution.
Yeah. We can use plastic. Not
so we can use the plastic also, and
untuk stock solution. Untuk stock solution.
And actually, this is for the culture media, but you're asking to us about the stock solution. So we can use the regular cap, or also we can use the plastic, and also
aluminum
foil. This aluminum foil.
For EDPA, you use aluminum foil. You have to wrap.
I think it's very sensitive to the light, right? Yeah, this one is sensitive to the light, so we need to cover it with aluminum foil. So for the cap, if you have the regular cap, I think it's okay.
Okay, yeah.
And if you don't have, we can use the plastic as well.
Okay, Miss Sangeeta.
Okay, there's another question from Tan Thuy Duong. I believe she or he from Vietnam. After preparing tissue culture, have to wait at least 1 week to check contamination of bacteria before making tissue culture.
Yes, Mr. Tan Thuy Duong. Yes, you have to wait at least 1 week to check the media before you put the explant inside. Yes. To make sure the media not contaminate with bacteria or fungus. Okay.
Okay. Miss Sun. Okay, thank you.
Yeah, thank you.
Is there any participant who would like to address question or feedback verbally, or maybe please type your question or answer on the Q&A session before we are heading to the next presentation. Okay, I think no further questions. Anyway, please take note that we will share the contact of the trainer so that you can have further consultation or maybe further discussion with the trainers outside of this event. So maybe thank you very much, Ms. Sani Hanifah, for the interesting presentation and for your effort to prepare all of the materials for the demonstration. It is very interesting.